The overall goal of this procedure is to silence BRCA2 expression by transfection of siRNA. This is accomplished by first preparing the siRNA transfection reagent complexes. Next, a first round of transfection is carried out by adding complexes dropwise to a monolayer of cells that are at 80% confluency. Then, a second round of siRNA transfection is performed using a higher siRNA to transfection reagent ratio. Finally, the cell monolayer is washed with ice-cold PBS, and the cells are lysed at 4 degrees Celsius with a detergent-based lysis buffer. Ultimately, Western blotting analysis is used to show BRCA2 silencing at the protein level.
Main advantage of this technique over existing methods is that it allows efficient gene silencing as well as optimal detection of high molecular weight proteins like the tumor suppressor BRCA2.
To begin, after growing human epithelial cell monolayers at 37 degrees Celsius and 5% carbon dioxide in a humidified chamber, remove the growth medium and use room temperature PBS to wash the cells. To detach the cells from the plate, add 2 milliliters of 0.25% trypsin, 0.53 millimolar EDTA solution, and incubate for 3 to 5 minutes, depending on the cell type. Neutralize the trypsin by adding 2 milliliters of complete growth medium containing 10% FBS before transferring the cells to a 15 milliliter tube. Place the tubes into a swinging bucket rotor and centrifuge at 320 to 330 Gs and 4 degrees Celsius for 3 minutes. Use 5 milliliters of antibiotic-free medium to resuspend the pellets before using a Burker chamber or automated counting system to count the cells. Then seed approximately